Hello and welcome. Today, we're diving into a technology that isn't just changing our world, it's literally rewriting the code of life itself. We're talking about biotechnology, and you'll see how it's shaking up everything from the medicine we take to the very food we eat.
So, let me just start with this pretty mind-blowing question. What if, instead of making medicine in some huge factory, we could program living cells to become tiny biological factories themselves? I mean, just imagine teaching a simple bacterium how to produce life-saving insulin, or engineering a crop so it can defend itself against pests. It sounds like science fiction, right?
Well, it's not. That right there is the core promise of biotechnology. And over the next few minutes, we're going to pull back the curtain and take a look at the scientific blueprint that's making all of this a reality.
But before we get into all the nuts and bolts, let's start with the big picture. What is biotechnology really?
Okay, so put simply, biotechnology is all about harnessing living things. We're talking everything from the smallest bacteria to complex plant cells, and using them to create products that make our lives better. And the key phrase here is for human welfare. The whole point is to create solutions for a better, healthier world.
And you might be surprised to find out this isn't some brand new idea. The term biotechnology was actually coined over 100 years ago, way back in 1919, by a Hungarian agricultural engineer named Károly Erecki. He's now considered the father of the whole field.
So, how does this all work?
Well, modern biotechnology really boils down to two core principles. You can think of them as the two essential chapters in the instruction manual for rewriting life.
On one hand, you've got genetic engineering. That's the art of modifying an organism's DNA to give it totally new abilities. And on the other hand, you've got bioprocess engineering. That's the science of taking that one modified cell you just created and scaling it up to produce things like vaccines on an industrial level. It's kind of like the difference between designing a blueprint and actually building the skyscraper.
So let's dive into that first principle, genetic engineering. I mean, how do you actually go about rewriting the source code of a living thing? Well, the key technique here is called recombinant DNA technology. And honestly, the cut and paste analogy is just perfect. Scientists can literally snip a useful gene out of one organism, say the human gene for insulin, and then paste it right into the DNA of a completely different organism, like a bacterium that grows super fast.
Now, I know it sounds incredibly complex, but scientists have really broken it down into a clear step-by-step playbook. So let's walk through it together.
It all kicks off with isolating the DNA that has the gene you're interested in. Then, using these things called molecular scissors, you snip out that specific gene. Now here comes the really clever part. You paste, or ligate, that gene into a vector. Just think of it as a biological delivery truck. That vector then carries the new gene into a host cell, like a bacterium. And from there, all you have to do is give that cell the right conditions to multiply, and boom, you get millions of copies. Finally, you harvest the product those cells are now programmed to make.
But wait a second. To do all this cutting and pasting, you need more than just one copy of a gene, right? So how in the world do scientists make millions of copies so they have enough to work with?
The answer is a process called DNA cloning. And just to be clear, we're not talking about cloning a whole sheep here. This is all about making millions and millions of identical copies of a single piece of DNA. And the key to all of this is that cloning vector we just mentioned. Turns out it's not just a delivery truck, it's a delivery truck with a built-in copy machine.
Okay, so for this molecular delivery truck to actually work, it needs a few essential features. First, it needs an origin of replication. That's basically the start copy button for the cell. Second, it needs a restriction site, a pre-marked spot that says cut here so you can paste in the new gene. And finally, a selectable marker. This part's brilliant. Think of it as a little tracking device that lets you easily find the cells that successfully took in the new DNA.
All right, so we've successfully engineered a single cell. That in itself is an amazing achievement. But how do you get from one cell in a lab to a medicine that can help millions of people? Well, that's where our second principle, bioprocess engineering, comes in and takes over.
And look, we're not just talking about a bigger petri dish. We're talking about scaling up to billions, even trillions of cells, all working together in perfect sync.
This is really where biology meets industrial engineering. Scientists use these massive, sterile tanks called bioreactors to create what's basically a five-star hotel for these cells to thrive in. The goal is simple: create the absolute perfect conditions for them to multiply and produce as much of that desired product as they possibly can.
And the products coming out of these bioreactors, they're things that have fundamentally changed our world. From the antibiotics we use to fight infections to the vaccines that prevent them in the first place, this process is right at the heart of modern medicine.
So we've gone from a single gene to global manufacturing. We've seen the how. Now let's really focus on the why. Why does all this matter so much?
This isn't just theory anymore. It's our reality. It's the insulin that keeps people with diabetes healthy. It's the promise of gene therapies that could one day cure diseases that are passed down through families. It's the development of crops that can survive a drought and help feed a growing planet. Biotechnology is already shaping our world in more ways than we can count.